Nucleotide synthesis, Denovo synthesis of purine ribonucleotide. Now this purine ring is assembled of various precursors such as glutamine, glycine, aspartate, N10, formyl tetrahydrofolate, then carbon dioxide. Okay, it means like atoms of these precursor make your purine ring. Okay, that is what it means. So this is how purine looks like, right? This is the structure of purine. Now as you can see that this nitrogen which is present at the first position, it is supplied by aspartate. Okay, this nitrogen came from the aspartate. Similarly, this carbon which is present at the second position came from this precursor, that is N10 formyl tetrahydrofolate. Okay, then this carbon which is present at the sixth position came from the carbon dioxide. After that, this carbon that is present at fourth, fifth, and this nitrogen which is present at seventh position, these three atoms came from the glycine. After that, this carbon which is present at the eighth position came from N10 formyl tetrahydrofolate. So this N10 formyl tetrahydrofolate is giving two carbons, with one carbon is present at 8th position and the second carbon is present at the second position. Okay, and after that these two nitrogen present at the third and ninth position come from glutamine. Okay, so as you can see, the atoms of all these precursor make your purine ring. Now the de novo synthesis of purine begins with simple starting materials such as amino acids and bicarbonate. Now unlike the pyrimidine biosynthesis, in case of purine biosynthesis, the purine bases are assembled onto the ribose ring. Okay, on ribose ring there is assembly and then the purine base is produced. Okay, that is how the synthesis of purine base takes place. Okay, it is assembled onto the ribose ring. Now this de novo purine biosynthesis, like pyrimidine biosynthesis, requires PRP. Okay, now in case of purines, this PRPP serve as foundation onto which bases are constructed. Okay, PRPP is used as a foundation and on this foundation bases are constructed, and that is how your purine biosynthesis take place. Okay, so yeah, pyrimidine biosynthesis and purine biosynthesis, both of them requires PRPP. That's the point. The initial step, okay, initial committed step in the purine biosynthesis is the displacement of the pyrophosphate by ammonia. Okay, when you displace the pyrophosphate by ammonia, then there is production of phosphoribosyl 1-amine. Okay, so in case of purine biosynthesis, there is no need of pre-assembled base. Okay, there is no formation of pre-assembled base and then that base will be attached to ribose. That is not how it takes place in case of purine biosynthesis. In case of purine biosynthesis, it goes step by step, and the first step is the displacement of the pyrophosphate by ammonia. Okay, so in first step, at the end of the first step, you will be having phosphoribosyl 1-amine. So this is how the reaction looks like. Okay, this is PRP. It is getting converted into 5-phosphoribosyl 1-amine. Okay, and as you can see, this OPP is getting replaced by NH2. Okay, and in this reaction is catalyzed by glutamine PRPP amidotransperases, and in this reaction the glycine is getting converted into glutamate. Okay, in this reaction glycine is getting converted into glutamate, and that is why there is supply of this amino group. So yeah, PRP is getting converted into 5-phosphoribosyl 1-amine. Now, to prevent wasteful hydrolysis of either of the substrate, that is PRP and glutamine, this enzyme amidotransferase remain active or show its active configuration only when it binds with both the substrate, that is PRPP and glutamine. Okay, so when amidotransferases bind with this both of the substrate, at that time only it shows its activity. Okay, and this mechanism is preventing the wasteful hydrolysis of either of the substrate. Now this enzyme's activity, that is the activity of amidotransferase, okay, is inhibited by glutamine analog such as azathioprine, and that is the reason why this azathioprine is used as an anti-tumor agent. Okay, this glutamine analog is used as an anti-tumor agent because it inhibits the activity of the enzyme amidotransferase. Now the addition of glycine followed by formylation, amination and ring closure yields 5-amino-imidazole ribonucleotide. Okay, this thing is produced by the addition of glycine, then formylation, amination and at last ring closure. Now this AMP and GMP are formed from IMP. Okay, IMP give rise to AMP and GMP both. Now let's see in detail how de novo purine biosynthesis takes place. Now carboxylate group of the glycine residue is activated by phosphorylation. Okay, in the first step of the de novo purine biosynthesis, the carboxylate group is activated, and then this carboxylate group is coupled with the amino group of 5-phosphoribosyl 1-amine. Okay, so this carboxylate group is activated and it is coupled with the amino group. That is the first step in the de novo purine biosynthesis. Now, because of that, the new amide bond is formed and the amino group of the glycine is free to act as a nucleophile in the next step. Okay, so in the next step, the glycine amino group will act as a nucleophile. And why this is happening? Because new amide bond is formed. So this is how the reaction looks like. 5-phosphoribosyl 1-amine is getting converted into glycinamide ribonucleotide. Okay, and there you can see that this NH2 here is getting converted into this NH and this group. Okay, so this is what is happening in this step, and this step is catalyzed by synthetase glycine, and in this step 1 ATP molecule is being consumed. Okay, so this is the first step in the de novo purine biosynthesis process. Okay, 5-phosphoribosyl 1-amine is getting converted into glycinamide ribonucleotide. Now in the second step, the formate is activated and then added to the amino group, and because of this there is formation of formyl glycinamide ribonucleotide. Okay, how in the second step there is formation of formyl glycinamide ribonucleotide. How this formation takes place? Well, the formate is activated and then added to the amino group. Okay, this is what is happening in the second step. Now some organisms uses two distinct enzymes to catalyze this particular step. Okay, in some organisms there are two enzymes which is producing your formyl glycinamide ribonucleotide. So one enzyme out of those two enzymes transfers the formyl group from the N10 formyl tetrahydrofolate. Okay, it takes out the formate from this compound, and the other enzyme then activates this formate, okay, which is obtained from this compound into formyl phosphate, and then this formyl phosphate can be added directly to the glycine amino group. Okay, so that is how, by utilization of two different enzymes, this process is completed in some organisms. So this is how the second steps looks like. Okay, this glycinamide ribonucleotide is getting converted into formyl glycinamide ribonucleotide. Okay, this reaction is catalyzed by transformylase. As you can see that this NH2 is getting replaced by CHO. So this is what is happening in the second step of the purine biosynthesis, and in the process this N10 formyl THF is getting converted into THF. So this is the second step in the biosynthesis of purine, de novo biosynthesis of purine. The inner amide group is activated and then converted into amide by the addition of ammonia, which is derived from glutamine. Okay, so this is how it looks like. Okay, as you can see, there is this C double bond O, and in this compound there is C double bond NH. So this ammonia is added here, and this ammonia has derived from the glutamine, and that is the reason in this reaction glutamine gets converted into glutamate. Now in fourth step, the formyl glycinamide ribonucleotide cyclizes into a 5-membered imidazole ring, and this 5-membered imidazole ring is found in purines. Okay, so in fourth step there is formation of this 5-membered imidazole ring, which is found in the purines. Now this reaction, this cyclization reaction, is favorable thermodynamically even though an ATP molecule is consumed. But why this ATP molecule is consumed? To ensure the irreversibility. Okay, so this reaction, this cyclization reaction should be irreversible. That is the reason why one ATP molecule is consumed in this reaction, even though this reaction is favorable thermodynamically. So this is how that reaction looks like. Okay, there this structure is getting cyclized, and that is how the 5-amino imidazole ribonucleotide is getting formed, and in the process ATP is getting converted into ADP, and that is the reason why this reaction cannot go back. Okay, it is irreversible, and this reaction is catalyzed by synthase enzyme. Now in the fifth step, the bicarbonate is activated by phosphorylation, and then it is attacked by the exocyclic amino group. Okay, the activated bicarbonate is attacked by exocyclic amino group. Now the product of this reaction rearranges and it transfers the carboxylate group to the imidazole ring. Okay, so your imidazole ring receives a carboxylate group because of this reaction. Interestingly, mammals do not require ATP for this reaction to occur. Okay, this bicarbonate attaches directly to the exocyclic amino group, and then this bicarbonate is transferred to the imidazole ring. Okay, so this is what is happening in the fifth step. So this is how the steps looks like. Okay, as you can see, this bicarbonate group is added here, and this is how that structure becomes. So that is how this 5-amino imidazole ribonucleotide is getting converted into 4-carboxy 5-amino imidazole ribonucleotide, and this reaction is catalyzed by the enzyme carboxylase. Now this imidazole, in the sixth step, this imidazole carboxylate group is phosphorylated again, and the phosphate group is displaced by the amino group of the aspartate. Okay, so imidazole carboxylate group is phosphorylated and the phosphate group is displaced by the amino group. Thus, a six-step process links glycine, formate, ammonia, bicarbonate and aspartate. Okay, these all are linked by this six-step process of purine formation. Now, all these intermediate contain all atoms but two atoms that is necessary for purine ring formation. Okay, there are two atoms which are necessary for purine ring formation, and these intermediate do not contain that. Except those two atoms, they contain all the atoms. So that is the reason why these intermediates cannot form this purine ring, because they don't have those two atoms. So this is how the sixth step looks like. 4-carboxy-5-amino imidazole ribonucleotide gets converted into N-succinyl 5-amino imidazole 4-carboxamide ribonucleotide in the presence of synthase and aspartate, and in the process ATP is getting converted into ADP. Three more steps complete the ring construction. Now the fumarate, which is an intermediate of the citric acid cycle, is eliminated. Okay, fumarate is eliminated, and this leaves the nitrogen atom of the aspartate joined with the imidazole ring. Okay, nitrogen atom of the aspartate is joined with the imidazole ring. Why? Because fumarate is eliminated. Use of aspartate as amino group donor and the release of the fumarate are the reminiscent of the conversion of citrulline into arginine in urea cycle, and these steps are catalyzed by the homologous enzymes in the two pathways. Formyl group from the N10 formyl tetrahydrofolate is added to this nitrogen atom. Okay, nitrogen atom accepts this formyl group, and this leads to the formation of a final intermediate which then cyclizes, and when this final intermediate cyclizes, there is loss of water, and this results in the formation of the inocetate. Okay, inocetate is formed when the nitrogen atom accepts the formyl group and it cyclizes, and there is loss of water. That all leads to the formation of inocetate. So you start with N-succinylo 5-amino imidazole for 4-carboxamide ribonucleotide, and on this fumarase enzyme acts in the presence of fumarate, and this is formed, that is 5-amino imidazole 4-carboxamine ribonucleotide. And after this, transformylase enzyme acts on this compound, and this compound is formed, that is N-formyl amino imidazole 4-carboxamide ribonucleotide, and then synthase enzyme acts on this compound, which leads to the formation of inosine monophosphate IMP in the end.